So all of the magic happens in this gorgeous building behind me, which is called Leiden Center 1. Let's go on in, I'll give you guys a tour. Hi, my name is Leo. I am a gene editing research scientist here at Vertex within the gene editing department. The projects that I'm working on right now are different gene editing technologies. How can we make these things better? How can we make them more effective so that when they get to patients, they can really be transformative therapeutics? It starts from data, getting that data generated and kind of making sense of it. Just go right across here to my neighbor, just also my manager, Eric. Say hi. Within our gene editing programs, a lot of times we work with cell lines, we make edits into the genomes of these cell lines, and then we do assays to read them out and see if the edits have been successful.
I mean, this is really where the kind of engine of innovation happened, right?
We just kind of draw experiment ideas. It allows you to have a starting point for you to then identify the issues that you have and then try to solve them. So now that we've looked at some of the data from these cells and everything looks good, let's go ahead and go work on them in the lab. So now that I have my lab coat on, let me give you a tour of our main lab space. So this is where we have cells that are being cultured. These are human muscle cells in culture. So I put this onto our fun little microscope here. We're basically just looking at how these cells are doing, how the shapes of them, how dense they are, and we're just going to get our cells in there into the sterile hood. It's just like the human body, cells don't like to be invaded by bacteria or viruses. And so this instrument here is just a very safe zone for these cells to be in, always kind of replacing old media with new media containing nutrients. Sometimes we call this feeding the cells. So this here actually runs an optimized automated procedure that will get our cells into DNA. So I'm going to put this plate of samples into the deck here. This we will continue tomorrow, run them through another series of processes and assays, and then we can read out the results, and it'll be very interesting to see what we can see. Gene editing is a very complex field, and sometimes you can have these really great ideas and they don't really work out. It's always good to keep in the back of your head the next idea that we might have might be the one. And I think we have a very great culture that puts science and patients at the forefront of our thoughts every day. It's great to be able to work within this really collaborative group of brilliant people who are all focused and motivated to solve the problem. It really helps to have all of these different heads to kind of bounce ideas between. They really add to the whole picture that the group is looking at. Vertex explores the ability for gene editing to potentially become a curative therapy for many diseases. And if you can, through a single treatment, cure them of the disease, I think that's the dream for any scientist to be able to take part in. I hope you got a good glimpse into where I work, especially looking at this amazing building that houses a team of brilliant scientists that are going to tackle very important challenges in therapeutics.